Let's now talk about mRNA processing. Genes of eukaryotic cells are segments of DNA that are quote unquote interrupted by segments that do not code for amino acids. Heterogeneous nuclear RNA, in other words, hnRNA, is RNA that is produced when both introns and exons of eukaryotic extracellular DNA are transcribed. As we can see in figure 13.23, if you have a DNA molecule that it is undergoing transcription and you have exons, which are the segments of eukaryotic DNA molecules that codes for amino acids, and introns, which are the segment of eukaryotic DNA molecules that carries no codes for amino acids, are both transcribed, then the hnRNA has to undergo different enzyme catalyzed reactions to actually take out the introns, then splice them out, and all of a sudden the exons have to be joined together to actually produce what is necessary to produce proteins, which is going to be that mRNA molecule that specifically has only exons, which are going to code for amino acids. In order for mRNA to code for amino acids, that RNA molecule needs to be taken from the nucleus into the cytoplasm. Then, during the process of translation, let's understand that the RNA sequence is going to be, in a way, separated into codons. And understand that this separation doesn't specifically mean that it's all going to be taken apart. This means that the machinery, meaning the ribosomes, are going to be reading the RNA in three segments, in three nucleotide segments, specifically by looking at the nitrogenous bases. So these three nucleotide bases that are going to represent the code words that are going to bring amino acids in is what is known as a codon. When it comes to the amino acids, because these three-letter codes or codons can represent more than one amino acid, it is said that the genetic code is degenerate. We can see in figure 13.24 an example of an mRNA molecule. And to give you an example, here we have the different codons that represent different amino acids. It's interesting to see that when it comes to the genetic code, even though most amino acids are represented by more than one codon, the reverse is not true. You don't have a single codon that represents more than one amino acids. For example, to represent the amino acid methionine, we only have one codon, which is AUG. And we're going to see that in addition to these codons representing amino acids, we actually have specifically three stop codons. And these stop codons are actually going to be utilized in order to stop the process of translation. Let's actually look at this process in detail. During the process of translation, in other words, protein synthesis, we're going to have an initiation step. In this initiation step, we're going to have at the cytoplasm a ribosome, which is made out of protein and ribosomal RNA, that is going to consist of a large subunit and a small subunit, come together and meet the mRNA that came from the nucleus. We see that the mRNA is going to align first with the small ribosomal subunit, and that's why the small ribosomal subunit identified AUG, which is actually the initiating codons of many proteins. And that is placed specifically in what is called the P site of the small ribosomal unit. Then a tRNA that identifies AUG as methionine is going to bring in what is called N-formal methionine, and is going to form a hydrogen bonding event with the codon. So the anticodon in the tRNA hydrogen bonds to the codon in mRNA, and it brings the amino acid. Then, once this has occurred, the large subunit for the ribosome is going to bind the small subunit to complete what is called the initiation complex. You can see these three steps within the initiation of the polypeptide chain synthesis illustrated in figure 13.27. Next, this chain needs to be elongated. And in figure 13.28, we see the chemistry that occurs inside of the ribosome in order to elongate the formation of the protein that is directed by using the mRNA as a template. Once you have the initiation complex, with the first amino acid, which tends to be F-met, in other words, the N-formyl methionine. The A site within the ribosome identifies the next codon. The tRNA that actually has a anticodon that hydrogen bonds with the codon will bring the next amino acid. Understand that the amino acid that we see here, phenylalanine, is actually determined by the codon that is going to base pair with the tRNA. In essence, in order to make a peptide bond, what is going to occur is that the amino side of the amino acid that is sitting on the A site is going to bind to the carbonyl of the amino acid that is on the P site. Once the amidation has occurred, then we observe that the growing peptide is going to be placed on the A site. And then on the P side, we have an empty tRNA. The reaction that catalyzes the peptide formation is called a peptidyl transferase enzyme. And all of this actually happens in the ribosome. So the ribosome, in a way, is a complex molecule. After the elongation continues to happen, one of the things that happens is translocation. And in the translocation process, the whole ribosome moves one codon along the mRNA towards the 3' end. The empty tRNA is released from the P site, and the tRNA that attached to the peptide chain moves from the A site to the P site. Now, in order for this process to stop, we need a chain termination. And the chain termination overall basically establishes that once the ribosome continues reading and it encounters a stop codon, then the stop codon is going to basically message for bringing into the system what is called the release factor. Understand that the release factor does not encode for an amino acid. The release factor, in essence, what it's going to do is that it's going to catalyze the hydrolysis of the completed polypeptide chain from the final tRNA. Then the empty ribosome disassociates, and then it can bind to another strand of mRNA and once again begin the process of protein synthesis where it's needed. We can see in this image that some of this requires energy, but the energy that is utilized in this process is going to be another form of chemical energy called GTP. Let's now do a practice problem in which we're going to learn how to write the primary structure of the polypeptide produced during translation given an mRNA sequence. Online, you can actually find a genetic code table. And the one that you see illustrated on the right side of the slides can be easily obtained by Googling the genetic code table. I selected this one because it has a better descriptor of the codons, in which now we see the first letter, the second letter, and the third letter of the codon, and we have the three-letter codes for the amino acids. When it comes to solving such problem, all we have to do is utilize our table and write the amino acid three-letter codes or full words, depending on what the question is asking us, for the codon that we have represented in the RNA. So for example, whenever translation happens, the first thing that we need to find within our RNA sequence is going to be the methionine. Methionine, which is given right here, is always represented by AUG. So as we scan our sequence, we see that the very first amino acid that we have is methionine. Next, we see that we have a codon UUG. So what we have to say is, well, the first letter is going to be U, the second letter is going to be U, and then from there, we can actually, looking through the table, see that UUG is leucine. Let's look at what is the amino acid that comes from the codon GAG. In order to do this, we're going to do first letter G, next letter A, which is right here, and then we're going to look for GAG. In GAG, we see that that is the amino acid GLU, which that is glutamic acid. Let's move on to the next one, which is AAA. First letter A, second letter A. So AAA represents lysine. Next, we have GUA, which is going to be letter G, letter U, and letter A. So GUA is actually valine, three-letter code VAL. And then we have GGA. If we look at this, when we have GGA, that's glycine. And these are the amino acids that are present in this segment. Now, I want to point out that if in this segment, additionally, we had, let's say, a stop codon, let's say that if after that GGA we would have had a UAA, in your answer, you should not write stop codon. Stop codons are not part of the amino acid answer because they don't encode for amino acid. So if you are given a sequence that there is a stop codon, you're only going to write your answer up until the amino acid before the stop codon. Also remember that at times when it comes to format, you must write the first letter in your three-letter code from the amino acids as a capital letter. Now let's learn about DNA mutation. During the process of DNA replication, some errors could occur. It is estimated that on average, 1 in 1 billion of the different DNAs could be replicated incorrectly. As you can see, it is very low instances in which these errors happen, but it could occur. A mutation is going to be any change that results in an incorrect base sequence on DNA. If you have an incorrect base sequence of DNA, understand that this is going to have effects also in the transcription of the mutated DNA, and this could lead to an incorrect amino acid for the sequence, even a protein. If we have a normal gene that is going to be transcribed and then translated, and it leads to a protein, having a mutation not only could lead to the production of an abnormal protein, or it could lead to the production of no protein. Let's look at the different types of DNA mutations that can happen. There are three main types of mutations that result in translatable damage of DNA. They are called substitution, insertion, or deletions. Let's look at each of them via looking closely at figure 13.31. In figure 13.31, we have a normal segment of DNA here. We have its mRNA, and then we have the amino acids that will be generated from that mRNA. Let's say that, for example, we have a base substitution. And in the case of this base substitution, what we have is a single base is substituted for another base. In the example in 1331, let's say that this base pair, C, or this nucleotide, I should say, gets substituted by a T. When we look closely at how does this affect the amino acid, specifically in this instance, the substitution of this first nucleotide in a codon from C to T is going to change the amino acid, specifically from glutamic acid to lysine, because we can observe that at the level of mRNA, the mutation creates a codon that encodes for a different amino acid. In this case, the creation of this new amino acid creates a mutation. Substitutions not always create a mutation that leads to a formation of a brand new amino acid. Because, for example, even though it's not illustrated in here, if let's say that the mutation was specifically instead of that first amino acid it was here in the third amino acid. And if the third amino acid would have been changed specifically to a C, so I'm just going to write my other mutation. So if the mutation would have been that we go from CTT to CTC, CTC creates the mRNA GAG. And that GAG creates the amino acid, that's what AA is, GLU. So if the mutation would have happened at the last base pair, notice that this substitution does not lead to a different amino acid because we still have the same one. When that happens, that is called a silent mutation. And it is silent because even though a substitution happened, the same amino acid was produced. We also have base insertion mutations. In the case of base insertion mutations, what is going to happen is that now we are going to insert a base pair within our DNA. And when an insertion happens, meaning extra nucleotides are added to the DNA region, this is going to change the reading frame overall for the translation process. Because remember that DNA gets transcribed and basically those changes will be incorporated into the mRNA. And when you have insertions, since now the reading frame is different, that's why we see effects on the amino acids after the insertion occurred. We also could have specifically deletion mutations. In a deletion mutation, what is happening overall is that then a nucleotide is going to be deleted. And when that nucleotide gets deleted, in this case, it is just illustrating the deletion of the A. Like the base insertion mutation, the deletion mutation also changes the reading frame, and it often leads to a non-functional product. So out of these three different types of mutations, to be honest with you, the ones that are creating more detrimental effects at the level of the proteins that are observable is going to be insertions and deletions. Genetic mutations have been popularized in movies. And X-Men is a franchise in which the characters in this movie are mutants, and their genetic mutations actually gives them superpowers. Many genetic mutations lead to disease. And instead of showing you the diseases that could be created by genetic mutations, I want to give you just some interesting genetic mutations that have been observed in humans and animals. One of the examples of genetic mutations is blue eyes in humans. Blue eyes are actually caused by a genetic variation between a pair of genes that are called HERK2 and OCA2. That's why blue eyes can be inherited from generation to generation. Albinism, which is caused by a genetic mutation that is usually passed from parent to child, is a mutation that disrupts the production of melanin. This can happen in animals, as you can see here by the albino peacock, but this can also be produced in humans. Lastly, polydactyly is a condition in which a person is born with extra fingers, or it could also be extra toes. So you can see in this image that this man actually have 6 fingers on each hand. Understand that polydactyly may also result from genetic mutations or environmental causes, but it is important to note that regardless, it is a genetic mutation.